Good afternoon. Welcome to the seminar AI in Real Life this week. I want to mention that this is a special week. We have a session that is common to this seminar, CME 500, but also CS 522, that is AI in healthcare, obviously given the topic of the conversation today. So we're very pleased to have Professor Carlos Bustamante and Daphne Koller here with us. Daphne has been a professor at Stanford for 18 years. She was the first machine learning professor at Stanford some time ago. She's the founder, as you can see, the founder and CEO of Insitro, and obviously very much involved in healthcare and applications of AI in that space. Professor Koller, Dr. Koller, has received a number of awards in her life and very, very well known in the community. She received the PICAS Award, the Presidential Early Career Science for Engineers and Scientists, and she's a MacArthur Fellow. Professor Carlos Bustamante is a professor here at Stanford in the School of Medicine. Is also very, very well known. She has been awarded a number of recognitions, including a MacArthur Fellowship Award as well. So in a sense, we have a very, very distinguished guests here, and I'm looking forward to the conversation. I want to mention that we are going to collect questions in a little differently this time. So there are 4 students that will be walking around with cards. So please, if you have a question, mark it on the cards, and we'll pass them to Carlos for discussion afterwards. So without further ado, thank you for coming, and I'll leave it to Carlos to continue.
Awesome. Good afternoon, everybody. How's everybody doing? Excellent. So it's a real privilege and pleasure to be here with you, Daphne. You know, I've long admired your work, and I remember when I interviewed for a faculty job here, I was so nervous, right? Because I said, oh, they want you to meet Daphne Koller. I go, oh, geez, Louise, right? And so I come in, and she says to me, I just read a recent paper of yours. I have to ask you a couple questions. And I said, okay, right, I'm on the hot seat. And she says, on this paper where you're measuring like the spring constant of DNA, and I go, oh, geez, it's the other Carlos Bustamante. There's another guy at Berkeley who works on this stuff. And I said, I'm sorry to disappoint you. I'm the other Carlos. Oh, you're the human population geneticist. She goes, okay, then I have another question for you on this other paper that you wrote. And I was just blown away. It's a true story. She's a polymath. She's just covered so many areas, and it's just a privilege and pleasure to be with you. So I want to begin by asking, what are the big healthcare challenges and really tricky problems that you see AI addressing?
Thank you, Carlos. Is the mic on? Okay, great. So first of all, thank you for embarrassing me, and I now remember this meeting, and I'm very glad that we were able to attract you to join our faculty here at Stanford. And it's a great pleasure to be here, so thank you all for coming. So I think that healthcare is at a transition point right now in terms of the ability to collect incredible amounts of data. When I left Stanford in 2012, there were a big data set in biology or health was a couple hundred samples. You were really fortunate if you had a data set that had 1,000 samples. And now we're in a world where in multiple different data modalities, you can find data sets that have hundreds of thousands or millions or coming soon even more of different samples. And that ranges from some of the human cohort data sets like the UK Biobank or All of Us or FinBank, where it's, you have genetics, genotype and phenotype data for hundreds of thousands of individual humans at very detailed granularity, all the way through to molecular data sets for data sets like TCGA or GTEx contexts, up through data sets that are created via increasingly better and more disease-relevant in vitro systems that employ a range of different technologies, many of which, like CRISPR, have only come about in the last 3 to 5 years. And so I think we're really at the cusp of a data revolution in the life sciences that in other aspects of society has really driven forward the machine learning revolution. And so I think that we now have the opportunity in some ways before us to ask ourselves, what are the critical problems in the healthcare space for which machine learning can readily be applied? Now the data acquisition is no longer the bottleneck. Because I will say, up until the point that you have really interesting and meaningful amounts of data, it doesn't really much matter what machine learning algorithm you apply, they all perform about the same. And it's when the data gets large that you can really start to get creative about employing new ideas and new technologies. So if you ask what are some of the more interesting immediate applications of machine learning in healthcare, I would say it's in the area of diagnostics, because that's the space where it's very naturally, the problem is very naturally described as a machine learning problem. You have an input, which is some characterization of the patient, and the output is some diagnosis or prognosis or something like that. And there is a proliferation, if you will, or at least the potential for proliferation of training data because of previously assigned diagnosis via physicians, or even better, in some cases, there is an ascertained diagnosis that emerges from down the line longitudinal data regarding that patient, so you can tell with even greater certainty what the patient has. So that space, I think, is really primed for application of machine learning.
Can I interrupt you for a second?
Please.
Would you mind giving us an example, just to kind of hone it in, maybe something that may have affected some of the folks here or family members?
Yeah, so actually, I think that's a great question, and let me talk about what I consider to be one of the more interesting success trajectories of machine learning in the space of diagnostics, which is in the area of pathology. This one's close to my heart because one of the last papers I wrote before I left Stanford is arguably the first digital pathology paper. It was with an MD PhD advisee of mine, Andy Beck, who we together looked at cancer pathology images, and the question was, looking just at the same microscopy data that pathologists have looked at for 100 years, literally 100 years, can you make better prognostic predictions than you can using cancer grading? And the answer is, even using the relatively primitive machine learning technology that we were employing at the time, and even with a relatively small data set of a few hundred samples, you could outperform a pathologist hands down. And maybe even more interesting than that was the fact that the features that we were using, because we deliberately took like a data-driven, unbiased approach to the problem of, let's not just look at the same stuff that pathologists have been looking at, but let's quantitate everything you could imagine about the image. It turns out that the features that were most relevant were the ones that pathologists had never looked at, which were the ones of how the tumor interrelates with the stromal tissue that surrounds the tumor. And today, for those of you who are in the know-how on cancer immuno-oncology, that stromal tissue is called the tumor microenvironment. And it is the most significant prognostic factor for the success of the immune system in battling the cancer. So arguably, our unbiased machine learning-based approach, which had no notion of the immune system, was one of the first harbingers of the tumor microenvironment.
So just so I can understand, this was in breast cancer,
yes,
is that right?
This was in breast cancer.
And did the ability to do a better job in the grading affect treatment? Would you make different recommendations?
You know, you could have. We tried really hard from within an academic environment to push companies to adopt this technology, not because we wanted the measly royalties that they would have paid to Stanford, for which we would have gotten 5% or something, but because we really wanted the technology to benefit patients. And we found it very difficult to get engagement from companies on technology that there wasn't a strong in-house champion for, which is why Andy, who subsequently went to Harvard and was on
By the way, we tried to recruit him at Stanford. He had a job at Harvard, but I think he's now left to start his own company, right?
exactly was what I was getting to, which is he tried within Harvard to get companies to adopt even better versions of the technology that now use deep learning and are much better than the sort of simple, naive stuff that we did back then, was similarly unable to do that, and that's why he left Harvard and now has a company that does exactly that and is getting much better success. So I'm just going to add one like codicil to this story because I think it's kind of an interesting sort of arc of that, which is that we were doing somewhat better than a pathologist using our old technology. With the new technology, he was doing on the same tasks that pathologists are already solving, much better than pathologists. The newest paper that just came out a few weeks ago, neither Andy nor I were involved in that one, but I thought it was a really interesting tour de force, is a paper that looks at cancer pathology images, and by looking at the tumor cells is able to identify with reasonable accuracy, not awesome accuracy, but reasonable accuracy, what the mutation of the tumor is that is driving the tumor. Is it P53 or EGFR or MYC or whatever? And that is something that no pathologist can do at all. And so you've now exceeded human level performance using this technology, not just quantitatively, but qualitatively.
100%. So that's awesome. Those are great examples, particularly when it comes to the treatment of disease, right? So you have the patient present, you've got to make a diagnostic call, and from which different treatment decisions are made. I want to now turn to the issue of prevention. I've got a favorite example of mine on the prevention side, but I'd like to hear yours.
Okay, if you promise to.
I'll tell you mine if you tell me yours.
Okay.
So on the AI side, what do you think is some of the most exciting stuff on disease prevention?
So I think if you look, again, thinking about the diagnostic area, some of the work that companies like Grail and Freenome are doing, which is early detection of cancer before it becomes metastatic, before it spreads, I think are an incredibly promising line of work that really helps us treat cancer before it becomes untreatable. Because once you have metastatic disease, with a few cases where immuno-oncology works or HER2 inhibitors, it's nothing really works. So other than cutting it out. So I think that's one area. And as we start to think about some of the diseases that are becoming very pressing on us, like Alzheimer's, there is the growing recognition that by the time Alzheimer's becomes symptomatic, it's too late to treat it. The damage is too pervasive, and there's too much of this protein misfolding that in some sense is sort of contagious within the brain and sort of infects from one cell to the other. And so you really need to catch it when it's early. And one of the big barriers to having better Alzheimer's treatments is that we don't have early biomarkers of when Alzheimer's is about to emerge. So I think that's an area where we don't yet have an answer, but boy, would it be important if we did. So what's yours?
All right, so mine. So who here has heard of familial hypercholesterolemia? A couple of people. Well, you guys don't count. You guys are over in my lab. So a handful of you. All right, so...
Can I raise my hand?
Yes, of course. So 1 in 200 people suffers from a Mendelian form of just extraordinarily uncontrolled high cholesterol. These folks present often in their 40s, either with an early heart attack or with very advanced cardiovascular disease in the clinic. If you could identify them early on, and there's 3 major genes, and you put them on a statin from the time, say, they're teenagers, you avert all that heartache.
Oh.
And Stanford has just had a beautiful paper called Find FH, you can look it up, where they've done this. We've applied it in our hospital to sort of screen, find the FH folks, and tries to generalize. So I think that's definitely sort of frontline on the germline side. Anyone with a genetic condition, we should do a much better job of trying to identify, and where you can intervene early.
Yeah, and I think in the same spirit, there was a beautiful paper just about a month or 2 ago that talked about polygenic risk scores for things that are much more complex traits than just 3 genes. And it's like this was a risk score that was comprised of, I think, a couple million SNPs whose function is completely, by and large, totally not understood. And yet, the signature, just by incorporating all these together into a single number, is predictive at the level of a Mendelian disease. So odds ratios of 10 to 15, which is between the highest decile and lowest decile, which is, again, for some of these are treatable using
100%.
things like statins and such. And so this is an area where these early prevention can really make a difference to people's lives.
Well, and the other thing on the cardiovascular side is that, so 90% of the FH patients don't know they're FH patients, they're undiagnosed. And secondly, when they are prescribed the medication that will save their lives, PCSK9 inhibitors, they're often denied. 60 to 70% of them are denied precisely because you don't have a genetic diagnosis. So if you can use the signatures in the EHR to identify them, it's sort of a win-win for the system.
Yeah, totally.
All right, next question. So we're going to change tack a little bit. You've had an incredible career path, from a distinguished professor at Stanford to founder of Coursera to your work on aging to your most recent startup. How did you decide at each stage the different directions you were going to take?
Yeah. No, that's a good question. It probably wasn't nearly as guided as it might appear from the outside. The Coursera transition really was, in some ways, the emergence of an increasing amount of frustration, and partly from this cancer story that I told you a few moments ago, where I really wanted to make an impact on people's lives and tried really hard from within where I sat in academia to have that impact in ways that are more direct than training a bunch of amazing young people, whether it's in my classes or as advisees, and I wasn't able to do that. And so the Coursera thing emerged as a byproduct, an unintended byproduct almost, of the online education initiatives that I and others here at Stanford had put in place. And we just put out these courses in the fall of 2011, and suddenly we had 100,000 people in each one of those courses.
Insane. That is
and
so
it was
insane.
like, what? How did that happen? We didn't do marketing.
What's it like teaching 100,000 people?
I know. It's just crazy, right? And you think about the fact that even for this cancer project, even if we'd succeeded, it would have been years of FDA approval and so on. And this was like within a month, we had 100,000 people who were touched by this. And so to me, this was an opportunity to make an impact that I just couldn't pass up and go back to the lab and write some more papers. And so I took what was supposed to be a 2-year leave of absence and went to Coursera and firmly intended to come back at the end of 2 years. And then at the end of 2 years, it was so fragile. Startups often are very fragile at the age of 2.
They're like kids.
They're very much like kids. And so I just couldn't leave and come back to Stanford, and Stanford has a fairly rigid leave of absence policy, and they made me choose, so I did. And then I stayed at Coursera for another 2.5 years, a little bit longer, until the end of the summer of 2016, where I felt like it was pretty stable and on a good trajectory, and I wanted to come back to the core of who I was, which is machine learning and machine learning applied to the benefit of societally meaningful goals, which health had been sort of the area that I'd been most involved in, and I'd become addicted to having an impact. I couldn't come back and just spend more time writing papers. So at that point, I decided to stay in industry, and I thought that Calico, with its focus on aging, which is such an important societal problem, might be a good place to do something meaningful, and as being part of the Google umbrella could really be a place where machine learning could be well deployed. Turned out that what I wanted to do had the focus on aging didn't really align with the direction that I wanted to pursue in terms of the kind of platform that I wanted to build up. So took me 1.5 years to come to that conclusion, and decided that was, if I wanted to build that, I really needed to build it outside. And so left at the end of February of this year, and am now really trying to build this platform of how do we rethink drug development from the ground up if we have machine learning and the ability to build predictive models as a core technology in our tool belt. What are the key bottlenecks in an industry that has been on the constant decline from productivity for the last 40 or 50 years? Can we move the needle on that? I don't know how many of you have heard of Eroom's law. Eroom's law, if you think about it, is the inverse of Moore's law. Moore's law is how, in the technology space, as I'm sure you all know, we get a doubling of productivity year after year after year after year. Well, it's not doubling, it's every 18 months. But it's been an amazing trend that's lasted for 50 years. In the pharmaceutical industry, it's the inverse. Every year, productivity declines. 30 years ago, I think, it cost $100 million to bring a drug from inception to market. Now it's $2.5 billion to bring a drug from inception to market. And the reason for that is not that any single stage of the process for a given molecule has gotten that much more expensive. In fact, there's been, in some ways, increases in productivity for a number of those stages. So if anything, it would have gotten cheaper if you correct for inflation. It's the fact that most of the drugs that we put in every single phase fail. And the funnel has gotten progressively sort of wider and wider, narrowing down to a smaller and smaller number of things that make it to the next phase. And so this is clearly a failure of our ability to predict when looking at something at one stage what's going to succeed in the next stage. And so that might be, maybe, an area where building better predictive models might help.
Jeez Louise. Okay, so I got 3 follow-ups, one for each of the things that you talked about.
Okay.
So on the education side, if I could.
Yeah.
If we have a room full here of undergraduates, if an undergraduate wanted to pursue a career in the applications of AI in medicine, what would you recommend in terms of technical skills and background and maybe additional activities that they should be involved in?
So first of all, if you're even potentially contemplating that, let me strongly encourage you to consider it seriously. This is an area where, as someone who's now a hiring manager to that kind of skill set, it is such a strongly depleted skill set. For those of you who are statisticians, I think there is a very significant p-value on less than you would expect by chance at that intersection. And it is a skill set whose demand is growing tremendously as big data in biology becomes, or in health, becomes a reality. And finding people who can really bridge the gap between those two communities, who can communicate effectively in both health/biology on the one hand and computing/AI on the other, those people are in incredible demand. So if you're considering this is a really, really good career and will likely remain so and become even increasingly so over the coming years. So I would say take classes that span both disciplines, try and spend time in labs on both sides so that when you graduate, you can really communicate with both types of stakeholders, because I can tell you that if you're trained as one or the other and have never been in an environment where you really needed to communicate, it doesn't, I mean, these are people who are incredibly collaborative, thoughtful, smart, and you put like a bunch of computational people and biologists in the room together and ask them to talk to each other, and they might as well be talking Thai and Swahili to each other. It's just two completely different languages, completely different mindsets. So people who are at that intersection are going to be an invaluable bridge between those two communities. And trust me, you will have no lack of jobs.
100%. I agree. I endorse that message 100%. All right. Now let's talk about aging.
Okay.
Something that as we get older, we start to care more and more about.
I know.
Okay. So is there a future of medicine where the objective is going to be to stop aging of the body rather than the treatment of disease? Do you think that's reasonable? And is there any sort of reality to that in our lifetime?
Well, so I think stopping aging is probably an overly ambitious goal. I think that
Maybe a misspecified problem.
Yeah, it would be nice, but I don't know that that's a feasible approach. I haven't seen any biological case studies of that, so an existence proof remains to be had. Slowing aging, there is actually an existence proof. So if you want to slow your aging, people, eat healthier, eat less, exercise more, sleep more, don't smoke. All of these are ways to slow aging. I mean, and by that I don't just mean, you'll live longer because you'll have less heart attack and less cardiovascular disease. There are actually aging signatures in your body that will become slower if you do all those things. So I think that maybe that's a facetious answer to your question, Carlos, but it's not completely facetious because it's an existence proof.
Sure.
And if it's an existence proof, that begs the question of, can you reproduce that using chemicals for people who don't have the discipline to do that on their own. And I think that's not an unreasonable goal for us to aspire to if we could figure out the mechanisms by which these interventions allow the aging process to proceed.
I mean, certainly in animal models, right? The metformin work, the rapamycin work. It's interesting, right?
And caloric restriction works.
Right, which is mimicked by those, right? For those of us on the caloric restrictions, how I'm going to be honest, is that's difficult.
It is very difficult.
All right. Third question that got raised in our discussion about your tremendous career path. Computing and wet lab culture. Right?
Yes.
Mixing the wet and the dry lab. Tell us about that, right? Why are you so excited about that approach, and where do you see those synergies?
So this comes back to the point that I made a few moments ago about the value of being cross-disciplinarily trained, which I think is just, because it is just such a difficult thing to create those lines of communication. Most companies that I've seen, and most academic institutions, Carlos, really fail in having, in whatever department or whatever company, one culture that is really ascendant over the other, with the other being viewed as almost like a service organization. So to take an extreme perspective, which I'm not saying is universal, I know life scientists who think of, computational people as like the IT group. They're the ones who fix your laptops. And then I know...
And get you p-values and...
Get you p-values. And then I know of, similarly, computational
100%.
people who think of wet lab biologists as those folks who sit there and pipette all day, and it's all like manual labor. And, neither of these... Technicians. And neither of these characterizations is true. So those are like the extreme failure modes. But even for, once you move away from those extreme failure modes, one of the things that we've, I think, at least so far, been able to do at Insitro, which we strive to maintain, and we've seen it be successful, is if you put these two groups in a room and they're genuinely solving the problems together as opposed to a siloed solution, you come up with solutions that are much better than you could by having that separation. Because it turns out that some things are really hard to compute, but hey, you should have just told me. I could have built a reagent to that and I would have measured it for you, or the other way around. You can't measure it easily, but you can impute it. And so by working together, you end up with a solution that's really, the whole is considerably larger than the sum of the parts. And so for any of you who are thinking about going into a company that would benefit from that type of, expertise on both sides, really try and make sure that it is a true partnership of the two groups working together.
All right, my last question before we get to the techno-utopian Black Mirror part of the discussion. What's driving all the hype in AI and AI in healthcare today? What's behind it?
So let me start with the hype on AI, because I think that's sort of so much more ubiquitous even than the hype in healthcare. So AI really has come a tremendously long way in the last few years. And I've lived in my longer than I like to admit career through multiple iterations of the AI hype cycle, where AI was going to solve all the world's problems, and then it was a word that no one dared use for several years when the hype cycle sort of came down. And what makes this hype cycle different is that in this case, the value that's been provided in certain areas is real, fundamental and real and significant in a bunch of different applications. And therefore, while I think the hyperbole is still overstated relative to the reality, and it's fueled by people, honestly, who in most cases don't understand AI and are talking about killer robots who are going to take over our, our brains and such, and a lot of other nonsense, which has zero validity. And if you ask any respectable machine learning researcher, they will say that. even when you subtract all that hype away, there's still a lot of substance left. So I hope that the nadir of the hype cycle, when we get there, won't dismiss all the advancements that have come. Now, as to what's driving that, it's really the incredible amount of data that's been collected. So yes, it's better models, and yes, it's more compute power, but fundamentally, it's the increasing amount of data. Which I guess maybe brings me to were you going to ask me about the risks? Because I was going to ask about
yeah,
the risks.
you're blowing all my techno-utopian Black Mirror questions,
okay.
so just keep riffing.
Okay, so I'm just going to talk about what makes why the data that we have today has enabled these better machine learning models, and what are the sort of risks that are sometimes ignored, which is the current machine learning models, because of the availability of the data, we're able to construct models that are much, much richer than what we've been able to construct before. So previously, in the old generation, like the one where Andy and I did the computational pathologists, we had to sit there and laboriously define every single feature that the machine learning algorithm could use. Like when we talked about the stroma in the tissue, was like stroma in the tumor, it's like the average distance between the tumor cell and the closest stromal cell. Like we spent hours brainstorming. You don't do that anymore. You put all the data into your machine learning algorithm, and it automatically figures out what are the key features that are relevant in the data that it's looking at. And the reason that's important is because people are actually pretty bad at coming up with relevant features, where computers are actually really good at coming up with relevant features. So that's all great, and that's what's driven the forward progress, but it's also a two-edged sword. So I'm going to tell you one sort of apocryphal tale here that's important, because to me it epitomizes the risks here. This is a colleague of mine who spent most of his career trying to figure out from radiology images whether there is cancer in the patient or not, and used to use the old-fashioned machine learning techniques like the kind that we used before, and it was achieving kind of acceptable performance, and then moved to using the new deep learning methods that are so powerful, and all of a sudden performance just shot through the roof. So being a smart guy, he realized if something looks too good to be true, it probably is, and he sort of dug into the data and found that MRI machines leave an invisible watermark on the image that's basically the identifier of the MRI machine. And you have some MRI machines that sit at primary care centers, which is where most of the controls come from, and some that sit at tertiary care centers, which is where more of the cases come from. So amazingly, the machine learning was able to home in on that watermark and basically was using that to predict whether it was cancer or no cancer. So the lesson learned from this is that while these machine learning algorithms are unbelievably good at homing in on subtle signal, they're also unbelievably equally good at homing in on subtle artifacts. And so quality of the data becomes paramount as you start to use these machine learning algorithms.
Awesome. All right. So maybe we can now turn to some of the questions from our audience. And I'll say that they're kind of bucketed into questions about Insitro,
Okay.
questions around advice for students, and then some business recommendations for Insitro that we can talk about. Okay. So first question. What exactly are the prediction problems that Insitro is tracking? We may have some folks in the audience. Maybe your investors may not be clear on...
Well, my investors are actually clear. It's just that... Yeah, no, we're not really talking about that right now.
All right. Nice try. I like it, though. Nice try. Nice try.
But let's just say that there are plenty of places in the drug development pipeline that if you look at it, you wish you had a crystal ball that would tell you the answer. So you put a drug into a human. Will it exhibit liver toxicity? Boy, wouldn't you wish that you knew that. This seems like a very natural prediction question. In fact, it's one that's even been looked at by one or two pharma companies, not with the new tools and not with the right data sets perhaps, but it is a prediction problem that would certainly help avoid a lot of failures. So it's questions that we're like that we're looking at. Will this work? Will this exhibit certain toxicities? What are some of the failure modes that a drug might have that we can help avoid, and things like that?
Cool. All right. Now we've got more of an IoT question. All right, you mentioned you're organizing massive data sets in-house. What are the main challenges that you have with hardware? From a data perspective, what machines and sensors are the most important to think about?
Oh, that's a great question. And in fact, one of the things that we are doing at Insitro is building a data factory. So I mentioned a moment ago that data quality is paramount. So unlike a lot of companies that have decided to use as a shortcut the availability of lots of data out there that's in the public domain, we looked at those data sets and realized that most of them are not great and have all these artifacts and issues that we mentioned. So we're actually building a data factory in-house that is a fully automated robotic data factory that will create not cells, not proteins, not molecules, but just data to train machine learning algorithms. And given the importance of quality of data and the fact that biological entities like cells are extremely sensitive to everything, then coming to the IoT question, everything is tracked. Humidity, temperature, which batch of reagents is used, which iteration of the machine, which time of day, everything is recorded in the metadata that's associated with the sample. Which lineage the cell came from, because, as the cell's passage through from one generation to the next, they collect mutations. And if you find the mutation in generation 37, you want to know how far back you ought to go, and was it also there in generation 32 and 21 or not? And so all of that is tracked so that you can figure out what it is that you need to try and correct for in terms of batch effects and artifacts, so you don't end up with that watermark problem that we talked about before.
Awesome, awesome. All right, a question from a budding venture capitalist, I think. So if you want immediate impact, why not focus your CS expertise on repurposing already approved drugs? There are many such examples: COX-2 inhibitors in depression, valacyclovir in Alzheimer's, and so on.
So yes, from a budding venture capitalist. So there's a number. So if you want there's a number of different answers to this question. One is the pragmatic one, which is that if you're repurposing someone else's drug, on which they hold the patent, then you are basically at their mercy in terms of how much they're willing to give you for whatever it is that you can tell them for telling them that their drug is going to work for another indication. So that's an issue right there. If you're doing it for a drug that's off patent, your situation is much worse, because then
where's the money to be made?
The money to be made. So that's one answer. It's not, I think, the most interesting answer. I think the more interesting answer is that I think the most important diseases and the most important advancements are to be made using new chemical entities against diseases for which none of the current treatments exist. And so I think that it's not to say that as we develop some of our predictive models, if it emerges that an existing drug turns out to be the right treatment for a disease, obviously we won't throw out that information just for the heck of it. But I think you need to build out the pipeline on a more firm foundation than on just existing drugs.
Awesome question. And I should admit, I editorialized that from the budding venture capitalist. Maybe they're not. It just seemed like the question you might get. All right. Two additional great questions. How might we incentivize industry to share data, open access?
That's a good question. I think there is a growing willingness to share certain types of data pre-competitively. So there's some discussions, there's the ATOM consortium, which several pharma companies are part of. It becomes more difficult as you move up the food chain. Sharing clinical trial data has huge implications on privacy and such that make it harder. Even tech companies where the data doesn't have the same level of sensitivity as healthcare data are not eager to share the data that they have. So I don't know. Honestly, I think if you really want to be blunt about this, I think it's fair to say that data is the oil of the 21st century, right. And you don't find people willing to just share that with everyone because it is what the company in many cases is based off of, which is why when people ask me today, hey, I'm a budding machine learning researcher, where can I do the most impactful work? I tell them, if you're interested in application as opposed to theoretical constructs, you might be better off doing that in industry, because you will have access to more data and much, much better computational resources.
So you just blew my other question,
Oh.
by the way.
Oh.
I feel like, you were...
By telepathy?
Yeah, by telepathy, you figured it out.
Sorry.
I agree with you on the open access and pre-competitive. It's neat to see, like, the UK Biobank is now receiving funding from Regeneron and AbbVie and others to generate that kind of data at scale, but I agree it's a thorny issue.
Yeah, I think human data is one where people can have control in some sense, or hopefully will be able to have control over the sharing of their own data, so that they can ask, as part of the stipulations of the participation in the UK Biobank is that the data be universally shared with qualified researchers. I think that some of the emerging consortia have a similar constraint, and one of, I guess, the promises, I was hoping not to have a session where we didn't use the word blockchain.
Dude, that was my next point, by the way. That's where we were going next, either way. So, because you mentioned data is the new oil, so who controls the mining rights? What do you think about the blockchain models for you to remonetize your data stream?
Might as well. Okay.
Let's roll.
Okay. So I think at that point there will be the ability to control the data. I would say maybe, and I'm far from an expert on blockchain, I want that clear from the start. I don't know that this will turn out to be a meaningful revenue stream for individuals. Yeah. I think this will be a much more significant aspect in terms of letting people feel like they have control over the use of their data and be able to share it in a way that is not as privacy revealing, I guess, or whatever, as currently happens when you just give your data to a company. But I don't expect this to turn into a huge revenue stream for any of you, sorry about that.
So our universal basic income isn't going to be coming from our compounding data assets?
No, do you think so? I mean, you're the DNA expert, Carlos. What is the value of a single person's DNA?
With exponentially growing data, it gets smaller and smaller. Exactly. Except that if you're in the tail of the distribution, then it might be gangbuster. I would say the coolest opportunities I see are actually in what I would call nonprofit drug collectives, just to choose something crazy. But the idea that people would come together in rare disease groups and try to use collective bargaining power of the data to improve their own access to it down the road, or to maybe even, if we think about, say, making a monoclonal antibody, right? That's not something that's so difficult that if you had the indications, you couldn't kind of bootstrap it and make it happen, and then, for those that would benefit directly from it, give it to them at cost plus a little bit, and then for the rest of us that would benefit, say PCSK9 inhibitors. Could you have developed PCSK9 inhibitors, made them available to FH people at cut rate, and then for the rest of us that need to take PCSK9 inhibitors because the Lipitor stopped working, we'd pay a different list price. I don't know.
I think that's an interesting model. I'm going to push back on the development of a monoclonal antibody is not that difficult, not because fundamentally you're wrong in that, sure, the creation of the DNA sequence that corresponds to a monoclonal antibody isn't that difficult. It's the everything that comes afterwards in terms of everything from developability of that thing in the first instance through to the clinical trials,
the
manufacturing, the CMC. I mean, there is a lot that goes into a drug long after you identify the target or even the chemical entity.
Oh, 100%. Yeah. I mean, and this is, it'll be interesting to see. The small molecules are sort of similar, right? In some sense, they sometimes have easier manufacturing, but not necessarily.
But then they also have a lot more off-target effects, and so the clinical. I mean, there's.
So that's why we need Yelp for drugs, right? Just go to safety, right? You go to phase 1, you prove safety, let whoever wants to pay for it, and follow and see what happens. Maybe in Gottlieb, that you might be able to get some of that through.
Even Gottlieb, I don't think has. No? I mean, and I'm a big fan, by the way, of our FDA commissioner.
Me too, by the way. I think he's unbelievable.
Unbelievable. Like such. like, it's rare to see a government administrator of this level of thought and forward thinking. Yeah, but anyway,
And
I don't
have
think
a
he's
drug
going-
company,
yeah.
but I think the guy is incredible.
Yeah, I mean, what he's done, if taking drug companies aside, what he's done with approval of devices is unprecedented. So for those of you who don't know this, up until a recent regulation, every time you pushed out a new version of software to a device, you needed to go and get that reapproved with the FDA, which basically means that these things became sort of these legacy systems that were never updated because the update process was so painful. And he basically he and the FDA created a process by which the process of pushing out software updates was FDA approved, which means that those of us who are software developers are used to pushing out software once a week or even once a day, now have a way in which that can happen for medical devices, which has never been possible before. And to me,
No, that's a
that
game
is
changer. I think
it's
it's a game
total
changer.
game changer. I think ultimately it might make its way to therapeutics as we get into personalized therapeutics. I mean, there's such amazing things that you can do if you're willing to take a fresh perspective on things, as Gottlieb has done with devices.
Well, for biologics, right, you can only regulate the process, right? Because
Yeah.
the outcome is somewhat, not stochastic, but, there's variation.
There's definitely variation.
All right. I guess the last question I have for you is Silicon Valley. Why, can we, or how can we, if you think we can, I think we can, continue to be driving innovation, especially now as we move from maybe consumer tech into healthcare?
Well, I think in that respect, Silicon Valley is really in a unique position. So when I talk to my Boston friends who consider themselves to be the bastion of biotech for, probably reasonable arguments, the one place that even the most sort of stalwart defenders of the Boston side of the world would concede is a place where Silicon Valley has the edge, is in that marriage of tech and biotech.
Yeah.
So they have amazing, pharma, amazing therapeutics companies. Even when you think about the technology of science, like new devices and new stuff like that, it's sort of, I would say, at parity maybe, but a place where we as an ecosystem have a unique advantage is in the ability to bring in what you would call tech, like machine learning, computing, and so on, into the healthcare space, and that's what's driven all of the tech giants sit on the West Coast, either here in Seattle, and I think that speaks to this unique ability that we have here. What's missing to really drive that forward is that
More bio.
Is more bio, is more people who really are the bilingual people. And so I think this really is the future of Silicon Valley. And I would just throw out one last little piece of this, which maybe speaks to some of your forward-looking projections. Science proceeds in epochs. If you look at the history of the last, 150 years, the late 1800s were the epoch of chemistry. It's when we understood the periodic table and we weren't doing alchemy anymore and trying to convert iron into gold, right? And then that emerged into the 1900s up to about 1950, into the epoch of physics, where we split the atom and fission and fusion and so on. And then in the 1950s, the era of computing began, which obviously relied on some of the earlier advancements in terms of silicon and microconductors and so on and so forth. And that led to the era of data in the 1990s, which was paralleled starting around there, maybe a little bit later, with the era of biology, with the sequencing of the human genome and microarrays and so on. The era that we're embarking on right now, the era that's going to last us for, I think, the next 30 to 50 years,
100%.
is that convergence between those two disciplines. So it's the era of data science in biology. That marriage is going to be the next epoch of science. So this is really, I think, a field that's really good to get into.
This is why I love talking to you. I mean, I couldn't be in more strong agreement, right? Like, I think about when we were in college, the biggest companies in the world were literally all oil companies,
Yeah.
right? And it had to do with the ability to bring engineering to, prediction and exploitation of resource. Then they became the big data companies, right? The Googles and the Amazons and Facebooks and Alphabets that today have so much cash on hand, they can't help but go into healthcare. It overtook retail as the #1 industry in this country. So I think you're 100% right. In 20 years, the biggest companies in the world will all be tech bio companies. It won't be biotech companies, they'll be tech bio
Exactly.
companies.
I agree. And I don't know that in a similar way to the fact that, the oil companies didn't turn into tech companies, I don't know that the tech companies
will turn
are
into
now
tech
going
bio.
to turn into tech bio companies.
Yeah,
So it's an
yeah.
opportunity to create a
100%,
next generation
100%,
of companies that are going to be the next generation of giants.
Innovator's dilemma, right? Solving that innovator's dilemma has continued to be an issue for us. Daphne, thank you. You've
Thank
been
you, Carlos.
incredible.
Thank
you,
you.
everybody.